Our topic focuses on the usage of gene therapy and bone marrow transplant for SCID patients. What is SCID? SCID is an acronym for Severe Combined Immunodeficiency. Because it's X-linked, it occurs mainly in males. It's an inherited disorder that affects the immune system. These newborns are prone to recurrent and persistent infections because they lack the necessary immune cells to fight off certain bacteria, viruses, and fungi. These newborns are predicted to have a lifespan of 1 year if a bone marrow donor is not found. What causes this disease? SCID is caused by a mutation in the SCID X1 gene located on the X chromosome. It is caused by mutations in the common receptor gamma chain, a subunit of several cytokine receptors. This commonly leads to a lack of T cells, functional B cells, and natural killer cells. These receptors sit in the plasma membrane of immune cells. When the gene is mutated, the receptors cannot form and are absent from immune cells. As a result, the immune cells cannot communicate with one another about invaders. Too few T and B cells are made to fight off the infection, and the body is left defenseless.
Here are some previous methods that are used to cure X-linked severe combined immunodeficiency. The first method is allogeneic hematopoietic stem cell transplantation. It is basically transplanting the hematopoietic stem cells from the matching donor to the recipient. The good side is that it has very high survival rate and there is no abnormal stem cell contamination while transplantation. However, it is very difficult to find the matching donor. Also, it can cause immune reaction problems. Another method is allogenic transplantation. This method requires collection of hematopoietic stem cells from the host and genetically modify this abnormal stem cells to become normal stem cells and transplant them back into the host. The good side is that it causes no immune reaction problems and it is easier to find the stem cells. However, the method of gene editing and culturing the stem cells is usually not very perfect, and there can be also contamination with the normal stem cells while transplantation. The last method is gene vector expression method. It is basically transfecting DNA vector with gamma chain gene, and after the transfection, this DNA vector can be integrated into the host genome. And after the integration, the gamma chain gene can be stably and highly expressed in the host. The good side is that it allows the formation of normal hematopoietic stem cells and its progenies. However, this method also causes genotoxicity, and this can lead to development of other diseases and side effects such as T-cell acute lymphoblastic leukemia.
Our study draws heavily on a previous study that was attempting to find a genetic solution to the disease SCID X1 as an alternative to bone marrow transplantation. Their method of repairing cells ex vivo and then retransplanting them could be adopted to our study. The objective of their study was to restore a single receptor with a small mutation in it in the HSC line. They did this by isolating somatic cells, reprogramming them into iPSCs, correcting the mutation using TALENs, and then differentiating the HSCs into the line they desired, which was in this case natural killer cells. You can see that the first step in that process was hematopoietic stem cells, which is what we are going for. The methodology in particular used for this was called TALENs, which are similar to CRISPR-Cas9. As opposed to CRISPR-Cas9, which requires a separate nuclease and guide RNA, TALENs have a nuclease fused onto the end of the DNA binding domain. Two proteins are required to make one cut, one on each side of the location of the cut. Designing custom DNA binding domains allows double-stranded breaks at arbitrary locations. Those custom DNA binding domains are obtained from the plant Xanthomus, a parasite which uses them to degrade the host's immune system.
We propose to utilize the methods described in the paper for HSCs. By editing HSCs, we can generate a downstream effect that will be beneficial in terms of forming a reliable pool of HSCs from the patient that can be used for autologous transplant without the need for culturing and worrying about the short lifespan of cells. Our application involves producing iPSC of the SCID patient and the use of TALENs specifically to HSC. We chose to direct our efforts to HSC in order to avoid limiting our pool of progenitor cells for transplantation that previous methods could not sustain. In order to expand our edited HSC population, GCSF or GMCSF can be used to stimulate blood loss that in effect will promote a surge of early progenitor cells to the peripheral blood. HSCs can be identified via the CD34 marker. Collected cells can then be transplanted to make up for the missing immune cells of SCID newborns, saving time, money, and endless culturing that would have otherwise been necessary in past applications.
Our process will be able to effectively cure patients suffering from SCID. The only side effects will be potential discomfort during HSC mobilization and the chance for aberrant genome modification.